So imagine that a body part suddenly rebels against you, leaching nutrients while contributing absolutely nothing to you. It's like a useless but needy person, a group member that suddenly does not work anymore yet still put their name in the credit even though they didn't put a single word in that report. Or even worse, they say that they worked harder, they demand a raise. Well, that is exactly what cancer cells are doing. However, their transformation into these so-called cancer cells, it does not happen in a single day. The previously kind and normal cell that helps your body experience such stress that accumulated until the breaking point where they think to themselves, well, if the body does not care about me, why should I live for it? They could not take it anymore. They turned sinister and rebel to live not for you, but for themselves. And they will do everything in their own right to grow by taking everything from you. They do not care about the rules. What is it for? Even to the point that they can invade from your body's immune system and even use it to fight back. Today, we will talk about one of the deadliest diseases and its rarely heard cause, cancer and epigenetic. So, for now, cancer is the second deadliest disease in the whole world, yet they're not an infectious disease. So, first of all, let's discuss what is actually cancer. Well, cancer cells are just like any of your normal cells in the body. They have the same DNA, which is the structure that contains our genes and shapes how we are now.
Wait, really?
So yeah, think of the DNA as a massive cookbook. Even though every cell has the same cookbook with the same instructions, a skin cell might only read the recipes calling for skin. Meanwhile, a heart cell will only read the recipes for heart. And on the other side, you have cancer cells. They somehow will read the recipes wrong or interpret them in a different way. It's like a recipe calling for a pinch of salt, but they will just dump a whole shaker of sugar instead. You still get something at the end, but it's definitely not what you wanted. Cancer cells will exploit this misread instruction to grow, divide, and get stronger. It is not out of malice though, it's just pure survival.
So, depend on this instruction, the cell can be different?
Exactly. In cancer cells, however, they don't have the correct blueprint. Their blueprint have different instructions than normal cells, or the cells interpret the instructions wrong. It's like reading a recipe, you are supposed to add salt and to make a fried egg, instead you add sugar. It will give an entirely different flavor, even with the smallest change, and the cancer cell will just exploit this to keep growing, dividing, and get stronger. It is not for malice, but it's just survival sake. This makes them very durable and resistant against the treatment we have, so even if you try to correct the instructions, they just keep doing it wrong.
I see. Does that relate to cancer specifically?
Yeah, so for cancer, these cells will straight up divide non-stop. That's their instruction that they keep doing, right? And that's because of their gene expression actually, that is heavily influenced by oncogenes, which are genes that will drive the cancer progression, and also they will suppress the tumor suppressor genes, which are the genes that will stop the cancer progression. Therefore, it will just stop the brakes. In most cancers, mutation or a change in the DNA sequence may occur in the oncogenes or tumor suppressor genes to keep the cells from dividing. It's like a car without brakes, the car will just keep going, right?
But can they fix those mutations?
Well, they actually can in theory, but for cancer specifically, unfortunately, the repair mechanism is broken. It's like you don't have the service for it. The repairman is just not here. So we actually have this thing called the cell cycle, and it appears like a compass. And as it circulates, the cell will undergo division. And in this cycle, they have checkpoints, like a marker saying that, oh, if they have this mutation, then the cell cannot progress to divide. However, in cases of a cancer cell, they just bypass the cycle and those checkpoints.
So, it's like trespassing?
A bit, if you think about it, yeah. They just blatantly divide. What's worse in this mutation is that they can just accumulate in your body, especially as you age. So, as your body becomes more susceptible to it, simple things like just the sun, the damage from the UV light, can cause this mutation to just accumulate.
Wow.
right? And these cancer cells, because they keep dividing, they will start stacking, and it can be seen as a swelling on your skin, right? Or just like a bump. But it's not actually cancer yet. Sometimes it can just be called the tumor stage, where the cells just grow abnormally and multiply. Cancer, on the other hand, is defined because they start spreading and invade other organs in like metastasis, for example. So they will start to invade and spread into the other organs, looking for a prey to survive yet unknown to them. It will also start killing the host, our body, which at the end of the day, if they are without a host, they will just die also.
So, the way they differentiate it, does it mean cancer has progression stage? Like they evolve?
Yeah, they do. Not exactly evolving, though it's more like there are four stages that we can identify. And simply speaking, the first two stages normally focus on the growth of the cancer. As the stages progresses, the tumor goes larger. Then at stage 3, the cancer will begin to spread into the surrounding tissues. And lastly, stage 4, the cancer has spread even into the distant part of your body and looking for a new prey. For like, imagine you have a cancer in your lungs, it will just suddenly appear on your stomach. So far we only talked about what is cancer in terms of mutation. But now let's talk about the actual topic of our podcast now, which is epigenetics. It might sound complicated, but honestly, it's just a fancy way of saying that your genes can be turned on or off without changing the DNA itself. Think of it like a light switch, for example, where the bulb is still here and it is not broken or not disconnected. But whether the room is bright or dark will depend only on the switch. So unlike mutations that changes your DNA at its core, epigenetic doesn't. Another way to imagine it is just picture your DNA as a giant library full of books. Each book is one gene. Now, every cell has the same library, but different book popularity. So not all the books are read equally. In one library, for example, you have like comic books that are super popular. In another, it's more like self-help books. Epigenetics is like the librarian who decides what goes on the shelf for popular books and what gets shoved to the back in the corner that no one reads. So one of the modifications that is very well known in epigenetic is DNA methylation. In essence, we can say that it's an attachment of a methyl group that generally silences the gene. So you can think of methylation as locking the popular books by the librarian that shows it in a hidden corner. The book still exists, but no one knows where it is. Therefore, it is silenced. So if the DNA is methylated, it is not expressed by the body.
So, is it like deleting the DNA?
Not really deleting. The DNA is still there. It is just not expressed. It is like silence. They don't have the right to speak.
But other than methylation, are there other ways of epigenetic modification?
Well, another common one is acetylation. It does the exact opposite of methylation. Imagine the librarian takes a book and they put it on the shelf for a popular book. Even shining spotlight on it, like you need to read them. This makes people want to like take it and just also read it. That's what acetylation does, turning on the gene to be more expressed. To picture it, your DNA is basically a super long, and to keep it neat, they need to wrap it around special proteins called histone, kind of like a thread around a spool. So acetylation will loosen the wrapping by adding an acetyl group, making the DNA easier to access and read. And when unwrapped, these genes are expressed.
So basically, methylation will turn the genes off and acetylation will turn them on.
Precisely. So now the big question is, how do changes in epigenetics actually promote cancer? But now that we understand how epigenetic works in general, it's actually pretty straightforward if you think about it. The usual cancer we know happens when mutation causes the gene that encodes proteins to be abnormally expressed, which then leads to various problems, right? It's, if we take the kombu from earlier, if you follow the recipe and add just the right amount of salt, the dish will taste perfect. But if you add way too much salt, it becomes inedible, while if you add too little, it's too bland. So yes, it is really just about having the right balance. Now, since epigenetic influence when and how often the gene is expressed, you can imagine it's like a restaurant deciding which recipes from the big cookbook is to be put on the menu. If certain recipe is promoted heavily, it will be ordered again and again, so that dish or protein gets made excessively. Likewise, if a recipe is hidden at the back of the shelf of the kitchen, maybe even locked inside the cabinet, the chef will hardly ever cook it, they just don't know how to do it. In that case, that protein doesn't get produced or made. And that's how cancer can develop even further. Now imagine if the restaurant decides to lock away the recipes for the safety dishes, the ones that keep balance in the kitchen and prevent chaos. Well, by silencing these protective recipes, the kitchen loses its quality control. Without these safeguards, mistakes go unnoticed, and over time the kitchen gets out of control. Dishes keep being made without orders, and that's what we call a cell that turns cancerous. They just do things without being asked.
I see. So it has the same effect as gene mutation, like regulating the protein levels, but it's not caused by mutation itself.
Yes, that's true actually. Do you know that our DNA is made up like of those four letters A, C, T, G?
Yeah, I think we learned about that in basic biology in high school, right? It acts like a code and it has specific pairings where A can only pair with T and C can only pair with G.
Yes, actually, these codes are called nucleotides actually. So here's the bit of a fun fact: the C for nucleotide is cytosine, and it is actually more unstable compared to the A, T, or G. Because of its chemical structure, cytosine often changes through a process called deamination. Think of it like a fragile ingredient like milk that spoils very easily. This is really important because the DNA code A, T, C, and G works like the letters in a recipe. Even one letter change in that recipe can turn the whole dish upside down and have a completely different result. And to make things even trickier, epigenetics can influence the process, making cytosine's instability even more dangerous than usual.
Wow, like how?
Well, studies found that usually cytosine is tagged with a chemical marker called methyl, and it will undergo something called deamination to turn it into a thymine. But if cytosine is untagged, or as we say, unmethylated, deamination will change it into uracil. But here's the big catch: uracil does not exist in human DNA, so the cell immediately recognizes it as a mistake and replaces it with something else, the right letter in general. But thymine does exist naturally in DNA. So when cytosine mutates into thymine, the cell gets confused. It's like opening your cookbook and suddenly seeing a recipe that says add sugar or salt. Both words look correct, so which one was the original? So instead of fixing it correctly, the cell hesitates and sometimes makes the wrong repair. That tiny change can eventually lead to a mutation.
Well, that's true, but doesn't our cell has a type of memory probably?
Yes, our cell do have a kind of memory, if you want to call it like that, but this type of changes is often just too confusing for the cell, as the newly changed T does not give an apparent "I'm wrong" signal. The wrong signal comes only from the wrong pairing, but the cell is confused which one is the correct one.
I see. And so these changes eventually can lead to mutation if the cell removes the actual correct nucleotide and leads to cancer.
Yes! Now that we've covered the basic concept of epigenetics and how they could influence cancer, let's take a closer look at the actual evidences of how epigenetic changes appear in cancer cells. Well, we have one study in 2017 that showed that in healthy cells, DNA methylation normally works like a control system, almost like a sticky note that says, "Do not use this recipe right now." It keeps unnecessary genes quiet, so the cell doesn't waste time or make something harmful. But in cancer cells, these sticky notes actually start disappearing. In other words, DNA methylation suddenly drops.
I see. So that means many of the genes that should stay silent are suddenly active?
Exactly. So without those off signals, the cell starts reading and expressing genes it normally shouldn't. Researchers call this hypomethylation or lack of methylation. And it's really not harmless at all. This loss of control can cause chromosomal instability. Think of it like a library where you normally have some books that are sealed off because they contain drafts, duplicates, or even confusing instructions. If those seals are removed, well, people might start copying or rearranging those books, creating errors or mixing up chapters. In the genome, this leads to unwanted DNA rearrangement, and sometimes entire sections of genes can get deleted by accident. So when that deletion affects important genes like tumor suppressors, the cell loses key instructions for controlling the growth.
Wait, could you please remind me about tumor suppressor again?
Okay, so there are two important groups of genes, tumor suppressors and oncogenes, right? Don't think of it this way. Tumor, like, you could just think of it this way. Tumor suppressors are like the safety instructions in the cookbook. Don't leave the oven on too long, or make sure the chicken is fully cooked. They keep the cooking process safe and under control so there's no after effect. Oncogenes, on the other hand, are like the go ahead recipes, like instructions for making a dish. They tell the cell, like, yes, it's time to cook, it's time to prepare this meal, now. Both, both of these sets of instructions are very important. You cannot run a kitchen without recipes, and you cannot ignore safety notes. The balance between them is what keeps the system healthy. So, in cancer cells, studies such as the one in 2022 have shown that the balance is lost. Because of the loss of methylation, the safety notes get ignored, and the recipes for a non-stop cooking are left wide open. The result? Well, the cell keeps cooking endlessly, preparing dish after dish after dish without a pause. This uncontrolled activity not only fuels growth, but also gives cells, the cancer cells, the ability to spread further.
I see. So this thing called epigenetic regulates but at the same time causes chaos not by only changing the original DNA sequence or cookbook word by word recipe, but on when and how it is used.
Yes, that's correct.
So, since epigenetics changes can cause cancer, it means there should be a treatment to target the epigenetics too, right?
Yeah, you're absolutely right. So, epigenetics work with chemical tags like methyl or acetyl groups, right? Remember? So it's like those sticky notes placed on top of your DNA. Some notes say read this, others say ignore this. Treatments can work by supplying these chemical groups or by blocking the enzymes that put them in the wrong place. For example, one anti-cancer drug called Vidaza works by preventing DNA from being tagged with metal groups. Normally, these metal groups lock and silence important protective gene like tumor suppressors, for example, which acts as a brake against the uncontrolled growth. By stopping the addition of these locks, Vidaza helps keeping those protective genes switched on, giving the cells a better chance to resist the turning cancerous.
I see, that's really interesting. Usually we hear about gene editing, but this is another way to control the genes.
Exactly, so epigenetics is fascinating because it gives us another level of control, right? So, the most exciting part is that your lifestyle choices can also influence how these tags are working. Think of your genes as master chefs in a kitchen. If you feed them fresh, healthy ingredients like broccoli, garlic, or green tea, they will cook up like protective meals that keep your cells in balance. But if all they ever get is processed food, it will be like you force a five-star chef to use only instant noodle seasoning. And over time, the cells and the recipe are getting distorted, and the restaurant, that is your body, will start running poorly. Science has even shown that compounds in food like green tea, polyphenols, or sulforaphane from like the broccoli can reactivate silent tumor suppressor genes, even in breast cancer cells that usually don't respond well to standard therapy. It's a reminder that what we eat is not just fuel, it's also information that your genes and your cells are listening to. Well, I think that's all for us today, and that's all I can share for now. In the end, epigenetics plays a major role in cancer, not by changing the DNA sequence itself, but by influencing how our genes are read, which then shapes your protein levels and then your cell behavior. We've only scratched the surface today. There are so many other forms of epigenetics modification that we haven't touched on yet, and this field is still very much evolving. Every year, we find new ways to help us better understand how these changes work and how we might design smarter, more targeted treatments. Who knows, maybe one day it will be your research that uncovers the next breakthrough in epigenetics and cancer therapy. So until then, thank you for joining me in this journey, and I'll see you next time.